All right, pre-AP biology. Today we're going to talk about mutations. Even though we've talked a little bit about mutations this year, we're going to kind of go back through what we've already talked about, and today we're going to take this a little bit further. So I want to go over a couple things here with you, just kind of a little bit of review. Um, we've been talking a lot about DNA, a lot about protein synthesis, how we how your cells take the DNA in the nucleus and eventually build a protein, right? I mean, that's kind of what we've been talking about the last couple of days. So I want to make sure I define for you what a gene is, because we're going to talk a lot about genetics the next couple of weeks. So I think it's kind of an important time to kind of focus on what this word means. A gene is just a piece or a segment of DNA, that contains instructions to build a specific protein. So we've been talking about genes. I just haven't really been using that word. We've been using DNA or pieces of DNA or segments of DNA or the sense strand on the DNA. I mean, all this is just instructions to build a protein. So a gene is just a specific instruction. It's like if you have a book full of recipes, right? And there's only one recipe in that book, for example, that might be for chocolate chip cookies, or for how to make meatloaf or something. I don't know. But every recipe is unique because you get a specific product from that. Well, every gene is unique because every gene codes for a different type of a protein. So we know that genes can be transcribed and they can be translated right into proteins, through that process of protein synthesis. Now, our proteins are what determines our traits. So, kind of indirectly, these genes control our traits because they control what proteins get built. And what proteins get built is what we physically express as our physical traits. So I think it's important to kind of understand this. All right, now we do know that mistakes can get made, right? Sometimes there's errors in DNA. We've talked about this earlier in the year. We know that mutations can happen. Mistakes can get made, and these mutations might be spontaneous, which means they just occur. They can be genetic or they can be inherited by your parents. So if there's a mistake, let's say on one of your mom's chromosomes or one of your dad's chromosomes, that mistake can be transferred to you, okay, through genetics. And then finally, they can be mutagenic. They can be caused by just environmental factors. We talked about, for example, skin cancer. Skin cancer would be caused by a mutagen, some type of radiation, in this case, ultraviolet radiation that can cause a mutation in your skin cell DNA. All right. Now, there's three different kind of categories of mutations. And this is the one that we've talked about before. We've talked about numerical mutations. These are mutations that affect whether or not you have 46 chromosomes. Some people have an extra chromosome, some people are missing a chromosome. Those are trisomies and monosomies. Those mistakes are made during meiosis. They're made when sperm cells and egg cells are produced, and sometimes those sperm or egg cells just are missing a chromosome or they have an extra chromosome. So that's where that occurs. We're not going to really talk about that today because we've kind of talked about it in the past. But numerical mutations, obviously, if an entire chromosome is affected, obviously multiple genes can be affected. So a lot of times when people have, like Down syndrome, or like Edwards syndrome or Klinefelter syndrome, it's not just one gene that's affected, it's all the genes on that chromosome. So if you have an extra 21st chromosome, right, you have a trisomy, there are a lot of genes on chromosome 21. So there are multiple things that are impacted. The word syndrome basically refers to a lot of complications. There's not just one specific thing wrong with people that have Down syndrome. There's multiple things that are wrong, but it all is due to them having an extra 21st chromosome. Now, another type of mutation is called a chromosomal mutation. Chromosomal mutations affect the structure of a chromosome. They don't cause like extra chromosomes or missing chromosomes. They affect the actual layout of all the genes on one specific chromosome. So again, here multiple genes are affected. I'm going to give you some examples of chromosomal mutations on the next slide. But chromosomal mutations and numerical mutations are not the same thing. Numerical is when you have an extra chromosome or are missing an entire chromosome. Chromosomal mutations are when one chromosome has several genes that are impacted on it, but it's not because there's an extra one or there's a missing one. All right. Then finally, we have gene mutations. Gene mutations, which we'll talk about today as well, they affect only one specific gene. So only one segment of DNA on a chromosome is impacted by a gene mutation. All right, so let's go back to chromosomal mutations here. And there, I want you to look at these different types. We're going to put myself. Let's just keep myself here for now. Anyway, when it comes to chromosomal mutations, there's four different varieties, four different types. So, for example, this first one is called a deletion. Now, if you look at this here, okay, this is a chromosome, and these letters here, A, B, C, D, E, all these letters, these represent genes that are on that chromosome. So every single letter, for example, A and B and C and D, these are all genes that are instructions to build a certain protein. So you can see here that there's multiple proteins that can get built with the instructions on this one chromosome. Well, if you have a deletion, there's a segment of that chromosome that is just lost. So in this case, you know, genes C, D, E, and F, they're just cut off for whatever reason. Okay. When that happens, obviously multiple genes are impacted. Okay. But again, that's just one chromosome. It doesn't affect the other ones. Um, it's not like there's a trisomy or a monosomy, but again, this is a chromosomal mutation because multiple genes are impacted. The second type is called translocation. Translocation happens when a piece of a chromosome, so example, this green chromosome here, okay, so you'll notice here that genes A through F, they were cut off, they were removed by, again, whatever reason, and they were transferred to another chromosome. So this blue chromosome is a completely different chromosome. Let's say, for example, this green chromosome is, I don't know, chromosome number 2, and the blue chromosome is chromosome number 17. So a piece of number 2 was cut off and put onto chromosome 17. That's not normal. Okay, that impacts, again, multiple genes, multiple proteins are impacted by that. So that's called a translocation. Duplication is kind of similar to translocation, except instead of the 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 genes being put on a completely different chromosome, they're actually put on the homologous chromosome. So, like for example, let's say this is chromosome number 2, and you have two of those. You have two chromosome 2s. So, a segment of this chromosome 2 was put onto the other chromosome number 2. So again, what that kind of results in is like the same genes, being read twice. So like certain proteins get built extra, and that's not normal. Okay, so that's a mutation. All right, but it's chromosomal because again, multiple genes are affected. We call that a duplication. Okay, make sure you understand translocation and duplication. Translocation is those genes are moved to a completely different chromosome, whereas in duplication they're moved to the same chromosome. It's just a homologous pair. Okay, so make sure we know that. And then finally we have inversion. Inversion is kind of easy. This one is where you take a segment of DNA and it's just kind of flipped upside down. It's kind of inverted. So the instructions kind of get read, I guess, kind of backwards. It's kind of like, if you're reading a recipe and all the words were flipped, upside down or backwards or something. So you can probably see how, proteins can get built incorrectly, if that occurs. So chromosomal mutations. All right. Make sure we understand the four different types. That's important. All right. Now, gene mutations are a little bit different because they only impact one gene. So, for example, here's a piece of DNA. Here's a gene, right? We know that gene will code for a specific protein to get built. Well, if there's a mistake in that one gene, either two things can happen. Either the protein won't get built right, so it'll be abnormal, or there won't be any protein built at all. Okay, so gene mutations are specific to specific genes. Okay, there's three types of gene mutations. One's called a substitution, one's called an insertion, and one is called a deletion. So let's take a look at all three of these. All right, first of all, a substitution. To kind of explain this, let's take a look at this sentence here. It says, the fat cat sat. So if you read that sentence, you can probably visualize in your head, right, some fat cat sitting down or something, whatever. But take a look at what would happen if we just substituted one letter for another, right? So instead of a C, we're going to, substitute it with an R. So we're just kind of changing one letter in the sentence. And that kind of, changes the whole meaning of the sentence. So now visualize in your head, a fat rat, sitting. So, I mean, it changes the meaning of the sentence. Well, this same type of thing happens with DNA. All right, so here's a DNA code, right? DNA sequence. So here's your triplets. Let's say that's the sense strand of DNA, and we know that DNA gets copied, right? mRNA copies it. So here's your mRNA codons, right? We did this the other day, kind of a review here. And then, of course, those codons can get translated into amino acids, right? In this case, methionine and arginine and threonine and leucine make up this particular amino acid sequence, right? And that's going to be your protein. But let's say that we have a substitution. Let's say that we have a mistake, right? So the mistake here, instead of this C, this cytosine, it's now a T. Let's see what impact that could have. So now we have our mRNA, right, that copies those instructions. And now take a look at your amino acids. So what this results is one amino acid instead of arginine. It should be arginine. It's now histidine. So that shows you right away that now that amino acid chain, which is part of our protein, is now incorrect. So that either leads to the protein getting built incorrectly, or maybe sometimes the cell won't build it at all if there's these incorrect instructions. Okay, so there's your substitution. There's one example of a gene mutation. An insertion. Let's go back to our sentence here, right? The fat cat sat. So now instead of substituting, we're just going to insert a letter. Now, when they insert one, what it does is it shifts all the other letters over. You'll notice here that we still see FAT, CAT, SA. We still see those letters, but they've all been pushed over one. Okay, that's what we call a frame shift. All right, an insertion is a type of gene mutation that shifts everything over. So in this case, you can probably imagine that multiple amino acids can be impacted by that. So again, here's our DNA, here's our mRNA, and here's our amino acids. So this is the normal sequence, right? So now let's say that we inserted a piece we inserted a nitrogen base right here between the T and the C. All right, so if we inserted that T in between these two letters, it pushes everything over. All right, so now when the mRNA copies this, right, this is what gets copied. And now take a look at your chain of amino acids. Now, every amino acid is different. Well, not everyone, but most of them are different from the ones that we had up here. So, an insertion can affect multiple amino acids, but again, it's the same result, right? You end up getting a protein that's not get built correctly, or it may not get built at all. So again, an insertion is again a type of gene mutation. Now, the last one is called a deletion. Now, a deletion is kind of like an insertion. It's just the opposite. So instead of inserting a base, we're going to remove one. So in this case, we're going to remove, the F. Okay, we're just going to take it out, and that shifts everything to the left. All right, so we're just kind of removing that one letter. But again, it's kind of a frame shift mutation, just like we had with an insertion. So instead of moving it to the right, it's going to move it back to the left. All right, so see how this impacts it. Right, here's your normal sequence. Right, and we're going to remove this T here. And if we do, everything after the T is going to move to the left. Okay, so this is what you end up getting. And then we copy that. And then here's our amino acid sequence. Okay, so you can see, how, just these little segments of DNA, they can easily be impacted just by one letter change, an insertion or a substitution or a deletion. All right, so the significance of mutations, you know, um, obviously, this can result in, certain genes being expressed incorrectly, you know, big mutations like chromosomal mutations and numerical mutations. They can end up in multiple genes, being impacted, multiple proteins being impacted. But I'll be honest, some mutations are neutral. Some mutations can occur and they don't really even impact the organism very well or very much. They just lead to variety. A really good example of this is tongue rolling. If you can roll your tongue like this, ooh, like that, okay. So if you can do that, okay, you're what we call a tongue roller. But the ability to do this, ooh, I'll do it again. All right. The ability to do that is actually caused by a gene. If you can do that, you have a certain gene, okay? If you can't do it, you also have the gene, but you have the mutated form of the gene. So if you're a non-tongue roller, okay, if you can't roll your tongue, you actually are a mutant. All right, you have a mutated piece of DNA that doesn't allow you to do that. It's kind of interesting. Another one, and we'll do this in class when we come back. Um, it's called PTC tasting. There's a certain chemical that I have these papers in class, and I'll give you guys one. But if you put it in your mouth, either you can taste it or you can't. It's one or the other. It's a certain type of chemical, and everybody's either a taster or a non-taster. But it's all caused by a gene. And if you can taste it, then you have this particular gene. But if you can't, you still have the gene. You just have the mutated version of it. All right. So it's kind of an interesting thing, but that's what genetics is all about. That's what your genes are. We all have these genes, but what forms of the genes do we have? Do we have I guess, the normal form of the gene, or do we have the mutated form of the gene? But I mean, some of these mutations are just kind of minor, like tongue rolling. I mean, who cares if you're a tongue roller or you're not a tongue roller? I mean, does it make any difference? Not really. Um, you know, it's just one of those things. It's kind of a neutral mutation. It doesn't really make a difference one way or the other. All right, guys, that's it for the notes for today. I will see you soon. Have a good day.